Here's a wild clinical fact for you. Harmless, everyday civilian bacteria, they do not cause cholera. It actually takes a silent, hidden viral infection to upgrade those civilian microbes into deadly biological weapons. In our last explainer, we looked at how animal viruses melt into eukaryotic cells, but if you're preparing for an allied health career, you absolutely must understand bacterial viruses too, because as you'll see, the line between a harmless microbe and a lethal patient infection is often drawn entirely by a virus. So today, we're going to break down exactly how this works by comparing the immediate, explosive destruction of the lytic cycle with the incredibly stealthy, silent integration of the lysogenic cycle. Here's our roadmap for this explainer. First, the lytic attack sequence, then lysogeny, then lysogenic conversion, and finally, we'll contrast latency with lysogeny. Section 1. The lytic attack sequence, a mechanical sabotage. To really wrap our heads around bacteriophages, I want you to picture a rigid, metallic injection drone, kind of like a sci-fi lunar lander. Now, there's a stark contrast here with our previous lesson. Animal viruses physically enter and uncoat inside the host. But bacteriophages follow a strict no uncoating rule. Because bacteria have a really tough outer cell wall, the phage drone literally anchors its leg struts on the outside and shoots a high-pressure mechanical harpoon straight through the steel hull. It just injects its raw genetic payload right inside, leaving its empty shell completely stranded on the outside. Let's walk through exactly how this rapid sabotage mission plays out in 6 sequential stages. Step 1 is adsorption. The mechanical drone uses its tail fibers to bind tightly to specific host receptors. Step 2 is penetration, which we just saw, that forceful injection of the viral DNA. Step 3 is synthesis. And man, this is the total takeover. The virus hijacks the host's ribosomes to frantically churn out viral proteins and duplicate its genome. Step 4 is assembly, where all those new capsomers snap together to build empty heads that get packed with the copied DNA. Step 5, maturation, sees the tail structures bolted right onto those loaded heads. And finally, Step 6, lysis. The newly built drones deploy special enzymes like lysozyme to dissolve the bacterial cell wall from the inside out. The cell just violently detonates, flooding the environment with hundreds of brand new raiders. Moving on to Section 2, lysogeny, the silent switch. Okay, so that lytic explosion is highly effective, but honestly, it's not the only strategy. Specialized lineages called temperate phages use a completely different, highly stealthy blueprint. This is called lysogeny. Think of lysogeny like a silent software patch. The temperate phage injects its DNA, just like before, but instead of destroying the host, it suppresses its destructive genes. It physically inserts and integrates itself directly into the host's single circular bacterial chromosome. It just goes deep into hiding. Once that viral DNA is fully integrated into the bacterial chromosome, we give it a new name, a prophage. Now, here is the really crucial point. This prophage is now trapped in a race of rapid reproduction. Bacteria reproduce constantly through binary fission. By hiding inside that master chromosome, the silent virus guarantees its blueprint is perfectly copied and passed down to literally 100% of the bacterial offspring every single time the cell divides. But what happens when that bacterial ship starts sinking? Well, that brings us to lysogenic induction. If the bacterial population is exposed to environmental stress, like damaging UV light or mutagenic chemicals, the silent prophage patch detects that its host's DNA is compromised. It acts as an ejection trigger. The viral DNA frantically cuts itself completely out of the bacterial chromosome, boots up its destructive lytic genes, and kicks the assembly line into absolute high gear to build escape drones before the bacterium collapses. Section 3. Clinical danger, lysogenic conversion. Now, let's look at why this matters so much for your clinical practice. We need to see how these silent viral infections actively create the deadly human pathogens you will encounter in the hospital. This phenomenon is called lysogenic conversion. When a prophage sits silently inside the bacterial chromosome, it isn't just taking up space. It frequently brings along unauthorized structural upgrades to the cell's genetic library. The virus often carries genes that encode for incredibly powerful exotoxins. So by simply being there, the virus turns a perfectly harmless civilian microbe into a lethal biological weapon factory. And this brilliantly illustrates that clinical reality. Notice these 3 notorious bacteria: Corynebacterium diphtheria, Vibrio cholera, and Clostridium botulinum. Without their viral infections, they would be completely harmless to us. It is actually the prophage that delivers the tox gene, forcing Corynebacterium to produce the suffocating diphtheria toxin. It's a temperate phage that gives Vibrio cholera the cholera toxin, driving life-threatening diarrhea. And it is a prophage blueprint that allows Clostridium botulinum to manufacture the incredibly potent neurotoxin that induces fatal flaccid paralysis. Section 4. Latency versus lysogeny contrasted. Let's bring this full circle now and structurally contrast the bacterial lysogeny we just covered with the animal virus latency concept from our previous lesson. First, we have to delineate the biological battlegrounds. Animal virus latency targets complex eukaryotic tissues. Think of human nerve cells or epithelial tissues. Lysogeny, on the other hand, strictly targets prokaryotic cells, exactly like our E. coli bacterium example. Next, let's compare their genetic states. In animal latency, viruses like herpes can hide out quietly as free-floating circular loops called episomes right inside the human nucleus. But in bacterial lysogeny, a prophage does not have that luxury. It must physically insert and integrate itself directly into the host bacterium's single circular chromosome. And because of that, their pacing is completely different. Animal viruses often sit completely dormant in stable, highly differentiated permanent cells, like human neurons that just do not undergo regular mitosis. Bacteriophages, however, are tied to active binary fission. The bacterial host continues to grow and divide at breakneck speed, forcing the viral blueprint to be replicated right alongside it. Finally, consider how they wake back up. Animal latency is often reactivated by systemic human triggers, things like fever, hormonal shifts, or general immunosuppression. But bacterial lysogeny is specifically triggered by direct, localized DNA damage, like UV radiation, which literally forces the prophage to physically cut itself out of the bacterial genome and start an attack. So I want to leave you with this. If a virus completely dictates a bacterium's clinical behavior, who is the real pathogen? Remember, viruses are the literal blueprints driving bacterial behavior and human disease. Mastering these mechanical blueprints is going to perfectly prepare you for your clinical coursework. Next, we are shifting into the lab to cover the specific techniques for cultivating and identifying animal viruses. So please go ahead and continue to the next explainer in the playlist.